Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Langerhans cell. These are dendritic cells which are key antigen presenting cells in the epidermis, particularly in the stratum spinosum layer. They are not only present in the skin, they are also present in oral mucosa, genital mucosa, lymph node, thymus, crypts of tonsils. Coming to its origin, embryology, from where they are derived, from hematopoietic stem cells, myeloid lineage, having dual origin. During embryo stage, from yolk sac, during fetal stage, from liver monocytes, mainly from liver monocytes only. During adult stage, they have the capacity to self-renew themselves. If depleted severely, they will be derived from bone marrow. Coming to its demonstration on light microscope using HNE staining, these cells will be visible as a clear cells. During processing, cytoplasm will be very pale. We can't appreciate dendrites, we can't appreciate internal organelles in HNE staining. They are called suprabasal clear cells. We have already seen they are present in stratum spinosum, that is the layer above stratum basal. Melanocytes are present in stratum basal, same visible like clear cells only. So melanocytes are named low level clear cells. Here Langerhans cells are high level clear cells with a coffee bean nucleus, perinuclear halo. This is the characteristic. This coffee bean nucleus also we can't appreciate in HNE staining. So we'll use special stains to appreciate its dendrites and structure, like metal impregnation staining with gold chloride, cobalt, zinc osmium iodide or osmium iodide. Enzyme histochemistry, ATPase positive, immunohistochemistry, CD1A, CD207, also known as Langerhans positive. This Langerhans is the most specific immunohistochemical marker of Langerhans cells. It is also S100 positive. S100 positivity is less specific and more sensitive because melanocytes also shows S100 positivity. Electron microscopic features. In electron microscopy, we can see all the internal organelles clearly. We can appreciate dendrites, coffee bean nucleus, which is ingrown, infolded, indented nucleus, Birbeck granules. They may be rod shaped or tennis racket shaped. Characteristic features, all those are. Coming to functions, it is a part of skin mononuclear phagocyte system. What constitutes a skin mononuclear phagocyte system along with Langerhans cells, dermal dendrocytes and macrophages. It participates both in innate and adaptive immunity. Using its dendrites, which extends towards the epidermal surface in a uniform manner, whenever antigen comes into contact with skin, they will trap these antigens using the dendrites. They will phagocytose, process, and then moves to present the antigen to naive T cell in lymph nodes. From skin, they will take the antigen, they will enter lymphatics, ultimately reaches the nearest lymph node. Why? Because they are antigen presenting cells. They have to present the antigens to naive T cells. Their naive T cells become activated, sensitized, and they will produce cytotoxic T cells and memory T cells. These memory T cells are responsible for adaptive immunity. In epidermis, they will show a unique motion in between keratinocytes. Why they will show this motion? To cover a wide area and to trap antigens efficiently. This unique motion is called as DSURH, dendritic surveillance extension and retraction cycling habitude. This Langerhans cells maintains balance between immunity and tolerance. If we have tolerance, we won't get autoimmune diseases. We will differentiate between a self antigen and a foreign antigen. When a self antigen like dead keratinocyte enters engulfed by the Langerhans cell, it will release cytokines like tumor growth factor beta, interleukin 10 and activates T regulatory cells, which will inhibit activation and chemotaxis of the T cells. So it won't allow unnecessary immunity against our own antigens, that is tolerance. It produces immunity. While producing immunity, it will release cytokines like interleukin 1, which will produce chemotaxis and activation of T cells, which will destroy the foreign antigen. Conditions where Langerhans cells are decreased are UV exposure in elderly, potent topical as well as systemic corticosteroids within warts, psoriatic plaque, and in HIV. In HIV, first it is one of the first cells to get attacked by the HIV virus. Virus enters Langerhans cell. Langerhans cell will go and present those virus to CD4 T cells. Then HIV infection will spread systematically to all the CD4 T cells. Where Langerhans cells will be increased in eczematous dermatitis like atopic dermatitis, contact dermatitis, drug-induced lichenoid eruption, lichen planus, Langerhans cell histiocytosis. Remember the channel name. Don't forget to like, share, and subscribe. Thank you. Hasta la vista. Das vidania.